All right, today we're going to unravel one of the biggest paradoxes inside every single living thing, DNA. You know it as the blueprint of life, right? The master code that makes you, well, you. But this code is hiding a physical secret that sounds completely impossible. So what is it? Let's get into it. Take a look at this number, 2.2 meters. That's about 7 feet. Now, get ready for this. That is the length of the DNA that's packed inside every single one of your cells. A cell so tiny you need a powerful microscope just to see it. So that brings us to the big question, the central mystery for today. How on earth do you solve a packaging problem like that? Seriously, how do you cram a 7-foot-long molecule into a space that's thousands of times smaller than the period at the end of a sentence? Well, that is the enigma we're going to uncoil right now. To really get our heads around this, we first have to appreciate just how wild this problem is. I mean, where does that 2-meter number even come from? Well, the math is just staggering. Your average human cell has about 6.6 times billion base pairs. Those are the letters of our genetic code. And the distance between each letter is a teeny tiny 0.34 nanometers. You multiply those two numbers together and boom, you get that incredible length. So right away, you know the answer isn't about shrinking the molecule. It has to be all about the structure, how this thing is built, and more importantly, how it's folded. So if structure is the key, we have to ask, what is the structure? Well, finding that answer was a thrilling scientific race, a real detective story to figure out the shape of the molecule of life itself. And this journey took almost a century. It started way back in 1869, when a scientist named Friedrich Miescher first found this weird acidic stuff in cell nuclei he called nuclein. Fast forward about 80 years, and Erwin Chargaff discovers a massive clue. In any living thing, the amount of A always equals T, and the amount of G always equals C. But the final, absolute knockout piece of evidence came from these incredible X-ray images taken by Rosalind Franklin and Maurice Wilkins. Those images gave James Watson and Francis Crick what they needed to finally crack the code in 1953. And what they discovered? Wow. It was an absolute masterpiece of natural design. So let's zoom in and take a look at the elegant structure they figured out. They came up with the double helix. You've all seen it, that iconic twisting ladder. It's beautiful, no doubt about it, but its real genius is in how it's constructed. So, the helix is made of 2 long strands, which are technically called polynucleotide chains. But for now, just think of them as the 2 long side rails of the ladder. So what are these rails and the rungs connecting them actually made of? To get that, we have to zoom in on the most basic building block of all, the nucleotide. You can think of it as one complete piece of the ladder kit. It's got 3 parts, a phosphate and a sugar. These link up to form the strong side rails, what we call the backbone. And then there's a nitrogenous base, which is the part that points inwards and forms 1/2 of a ladder rung. And those bases are the famous 4 letters of life, A, G, C, and T. But here's the thing, they're not all the same size. They come in 2 flavors. The bigger ones, called purines, that's A and G, and the smaller ones, called pyrimidines, that's C and T. And here is the really clever part. To keep the ladder's width perfectly even all the way down, a big one always has to pair up with a small one. A purine always pairs with a pyrimidine. Okay, so let's put it all together. You've got 2 strands that run in opposite directions. The backbone is just alternating sugar and phosphate molecules. And in the middle, adenine always pairs with thymine, and guanine always pairs with cytosine. This strict AT and GC pairing rule is brilliant because it keeps the distance between the 2 sides perfectly uniform for the entire 2.2-meter length. It makes for an incredibly stable and consistent structure. Okay, now that we get the elegant, super uniform structure of the DNA molecule, we can finally get back to our big question. How is it packaged so tightly? It is an absolute masterclass in biological engineering, and it happens in stages. First, the DNA helix itself is negatively charged, and it gets attracted to these positively charged proteins called histones. It's literally like thread wrapping around a spool. This DNA and spool unit is called a nucleosome. Then all of these little nucleosomes start to coil up together, forming something that looks like beads on a string. We call that a chromatin fiber. And finally, that fiber gets coiled and supercoiled again and again, condensing down into the compact chromosome shape we all recognize from textbooks. And check this out. This isn't a drawing. This is a real image from an electron microscope showing that beads on a string structure. You can literally see the DNA strand, the string, and the clusters of histone proteins, the beads. To go from a 2-meter-long problem to this elegant, organized structure, it's just an incredible feat of natural engineering. So we've solved the packaging problem. We have this perfectly stable, unbelievably dense library of information all tucked away. But a library is pretty useless if you can't read the books, right? So why go to all this trouble? What is the ultimate purpose of this code? Well, that brings us to the very foundation of how life works. This simple flow right here is what Francis Crick called the central dogma of molecular biology. The master code stored in DNA is transcribed into a temporary messenger molecule called RNA. That messenger then carries the instructions out of the nucleus where it gets translated into a protein. And proteins? Well, proteins do pretty much everything. They are the microscopic machines, the building blocks, and the chemical signals that literally build your body and keep it running every second of the day. And that really is the ultimate takeaway. This entire, impossibly long, beautifully packaged code exists for one simple reason: to provide the instructions for life. From a single cell to a complex human being, everything flows from this elegant process. And it really leaves you wondering, as we get better and better at reading this amazing code, what profound secrets about ourselves are we going to uncover next?